Hello, this is Suganti, Assistant Professor, Department of Biotechnology, Vistas, Chennai. In today's class, we are going to see the basic introduction about molecular biology and the genome organization of prokaryotes. First, we will see what is molecular biology. Molecular biology is a branch of biology that focuses on the study of biological process at the molecular level. So, this is one of the important discipline in biotechnology. This may focus on the study of the biological process, how the DNA is converted into RNA and RNA to protein. So all these processes at the molecular level. It involves the understanding of the structure and function of the biological macromolecules. Uh, so molecular biology start with the understanding of the structure and function of the biological macromolecules. We all know that we all know about the macromolecules, right? What are the macromolecules? So these are all like DNA, RNA, proteins. All these are macromolecules. So how here we are in this discipline, we are going to see about how this DNA, RNA, and protein contribute the regulation and execution of the cellular process. And also this molecular biologist plays an important role in understanding the mechanism of inheritance, gene expression and the regulation of the cellular functions. So the what are the main components involved in molecular biology? It's nothing but deoxyribonucleic acid and protein. So this is the combined subject of three different subjects. It's a combination of biochemistry, genetics, and cell biology. Now we'll see some basic history about the molecular biology. The term molecular biology was coined by an American scientist, Warren Weaver, in the year 1938. And we all know that Linus Pauling, he is the father of molecular biology. And we all know that the milestones of molecular biology, without DNA, there is no molecular biology. And we all know that the greatest model finding of Watson and Crick in 1953, and also the base studies of Maurice Wilkins and Rosalind Franklin. And in molecular biology, the transfer of genetic information from DNA to RNA and then to protein is called central dogma of molecular biology. This is very, very important in molecular biology. So all the other topics are related to this central dogma of molecular biology. This central dogma is coined by Francis Crick in 1958. So this theory, central dogma, states that DNA contains the instruction for making a protein, which are copied by the RNA. Then RNA uses the instruction to make a protein. So the constitutively, the gene is expressed in two steps. One is conversion of DNA to RNA through transcription. Then RNA translated to protein through translation mechanism. So by these two mechanism only, the genes are expressed. Now, before entering into the genome organization, we will see some basic, very basic things in the about the cell and DNA. Okay, uh, I am having one question: whether the DNA is only present in nucleus in all the cells. I think you all know that there are two types of cells. One is prokaryotic cell and another one is eukaryotic cell. In prokaryotic cell, the DNA is present as a nucleide material. So all the prokaryotes are a single cell. So the DNA is present as a nucleide material. But in case of eukaryotic cell, the DNA is not only present in the nucleus, it's also present in the chloroplast as a chloroplast DNA or plastid DNA, and another component called the DNA is present in mitochondria. So that DNA is called as a mitochondrial DNA. So in eukaryotic organism, mostly the DNA is present in three different compartments: nucleus, chloroplast, and mitochondria. So in this is the comparison of prokaryotic and eukaryotic location of the DNA. So in the molecular biology, we will start with the organization of the genetic material in prokaryotic and eukaryotic cell. First, we will go with the model organism like E. coli. In E. coli, how the genome is organized. That means the genome is nothing but DNA. How the DNA is located or present inside the prokaryotic cell. So before that, we must know what is genome, right? Genome is nothing but it is the complete set of genetic information in an organism. So this complete set, it will provide all the information the organism required to function. So in living organism, the genome is stored in long molecules of DNA called chromosomes. The study of genome is called as a genomics. And you must know some terms related to molecular biology, like what is DNA, what is genome, what is genetic material. Genetic material is nothing but DNA. It's the hereditary material present in the cell. It carries all the information specific to the particular organism. So the genetic material is nothing but DNA, deoxyribonucleic acid. And we all know that the basic introduction about DNA. DNA is the hereditary material in human and almost all living organisms. Most of the DNA is located inside the cell nucleus, and it's called as a blueprint of life. And the main role of DNA molecule is the long-term storage of the information. Now we'll see the how the genome is organized inside the prokaryotic cell. Okay, we have already seen about the eukaryotic genome organization. In this class, we'll see about the prokaryotic genome organization. We all know that bacteria are very small unicellular organisms that do not contain the nuclear envelope and mitochondria, endoplasmic reticulum, and mitotic apparatus and other nucleolus, etc. And we know that there is no cell division. It's the cells are divided fission. Uh, so this is a prokaryotic cells are unicellular, and also the organization, the cell also very simple when compared to the eukaryotic cell. The cellular organization of the prokaryotes are very simple. So automatically the genome organization also very simple when compared to the eukaryotic cell. In eukaryotic cell, the genome organization is very little complex and tedious, but in prokaryotic cell, the genome organization is very easy. Now we'll see how the genome are organized in the prokaryotic cell. Actually, the bacteria have a very rigid cell wall which surround their cytoplasmic membrane, and their cytoplasm contains the ribosomes, mesosomes, and several granular inclusions. About one-fifth of the cell volume is occupied by the DNA, the genetic material. These are all about the introduction about the prokaryotes. Now we'll move on to the organization. Now you must know uh, what is the characteristics of prokaryotic DNA. Okay, the eukaryotic DNA are usually very longer and it's a linear molecule. Here the prokaryotic cells have a single circular DNA molecules that contains nearly all the genetic information, and it's present in the cytoplasm as the nucleide material. So the how the prokaryotic DNA is organized inside the cell means the DNA molecule is extremely long and is compacted by a process called supercoiling. By the method of supercoiling, the DNA is wound each other in the inside the cytoplasm in the cytoplasm of the prokaryotic cell. This supercoiling is mainly done by the enzymes called topoisomerase. These are the enzymes that will give the supercoils. So what are these supercoils? Just to pack the DNA inside the cell. Okay, and also this enzyme also introduce the additional turns into the DNA double helix and cause the DNA strand to wind up on itself. Just for packing. So in eukaryotic cells, the DNA is packed by histone molecules, and here the topoisomerase enzyme it will help for the supercoiling and the turns to wind up on itself. How the topoisomerase act in the genome organization means it will act by breaking the DNA polynucleotide and relating the ends relative to each other. Then enzyme rejoin the ends by a process called positive supercoiling. There are two different supercoiling. One is positive supercoiling. That means one of the enzyme it will uh, um, what to say, it will break and relate the two ends related to each other, and another enzyme it will rejoin the end by positive supercoiling. They are telling about the turns in the DNA. And while doing the turns, it will also remove the coiling in a process called negative supercoiling. That means it will do the turns in the opposite direction. So likewise, the both by playing the positive supercoiling and negative supercoiling, the topoisomerase enzyme it will help for the DNA to pack inside the prokaryotic gene. And in some research papers, they have discussed that some of the proteins are mimic to the histone protein, and they have isolated from the prokaryotic cell. Uh, and they are suggesting that they may also help in the packing of the DNA into the compact structure. But this topoisomerase only the major enzyme is involved in the packing of the DNA in the prokaryotic cell. So in this picture, you can see the supercoiling of circular DNA molecule in the model organism E. coli. Here you can see the E. coli cell structure. E. coli containing the circular chromosome in an unfolded manner. Here, with the help of topoisomerase enzyme, the chromosomes are folding into loops. Here you can see the looping structure. And further, the topoisomerase will add the turns and supercoiling. The supercoils, you can see the supercoils, and also some of the enzyme it will form the nick. The like endonuclease enzyme it will form the nick, and there are the common process. But this is how the eukaryotic, sorry, not eukaryotic, prokaryotic DNA are packed inside the prokaryotic cell. Then next, so inside the genome, the genes are present. We'll see some important points. Almost all the genes are present in the bacterial chromosome. Some of the genes are present in the plasmid. Some catabolic genes are present in the plasmids, right? You know that plasmids are extra chromosomal DNA. It's also present in the prokaryotic cell, and most of the genes are required for the structure and function are present in the bacterial chromosome, and some of the catabolic genes are present in the plasmid DNA. And the total length of the total bases is around 4.6 million base pairs, and the E. coli chromosome is containing the 4,300 genes. And we know that lab copron and true copron, right? Some genes are arranged as a families, like lag genes, right? So this concept is called as operon, and these genes also encode some protein with related function and are related in a coordinated way. So this is about something the genes, the prokaryotic genes. Thank you.